Today the plan is to spend most of the time with prokaryotes. We'll finish up a few points about the history of life on Earth first. A couple of reminders. First test is on Monday. There'll be 30 multiple choice questions. All you need to bring is a photo ID and a pencil. We'll provide Scantrons. We remind you there's a review session tonight that will be run by the BioSci peer tutors in this room from 5 to 7. And remember, for any score posted on the course website in the gradebook, you always have one full week after posting to ask about any kind of problem, like why did I get a 0? I think it was supposed to be, 1 or whatever it is. So after a week has gone by, we'll just assume it's correct, and we'll no longer discuss any problems with the score. This is because we just can't practically deal with hundreds of questions at the very end of the quarter. So let's keep up on those. So as clicker scores, for example, are posted in the gradebook, take a look. If there are any problems with the clicker score specifically, then Larry Cabral is the individual to email with any questions. I was also asked to make an announcement in this particular section. The class before us has a quiz every single Monday. And so at the end of the quiz, then some of the people have come in. She's told me that there are some students that go ahead and start coming in, and that this is a bit disruptive. So if you wouldn't mind, please not entering the room until 2:50. The previous instructor would greatly appreciate that. So thank you. So let's go back to considering the history of history of life on the planet Earth. And again, just to remind you, the Earth is approximately 5.6 billion years old. I do expect you to actually know that a number. Why on earth would I want you to remember a number like that? Is because that then provides a landmark, a yardstick against which you can compare other numbers. So I think it's very important. I did point out that although geological time is divided into a rather overwhelming number of different subdivisions, you'll be responsible for a very small number. I'll identify those absolutely explicitly. So for example, I pointed out that time is divided into 4 eons. You will be responsible for knowing the name and approximate time of our own eon, which is the Phanerozoic. So if you hear the word Phanerozoic, I would expect you to think, yes, that's our eon. It's the most recent, it's the current eon. It's the 4th eon. When did it start? On the order of about 500 million years before present, or million years ago. Geologists are very fond of acronyms such as this. MYBP would be million years before present, MYA, million years ago, whatever. BP would be billion years. So anyway, the Phanerozoic eon then started about 500 million years ago. Yes, it says 542. I don't expect you to know to that level of accuracy. 500 million years is plenty. So of course that's, what, about half a billion. All right, so that gives you an idea of what percentage of the entire history of the planet Earth are we talking about when we consider the final eon. Now, last time we considered how long it took for, best estimates for about how long it took before life evolved on the planet at all. And we considered one step out of a number of steps that would be necessary to start from an abiotic, an Earth with absolutely no living creatures on it whatsoever, to cellular life forms. We started with just the step 1, which is formation of organic molecules. There are obviously a lot of other steps, and we won't have time to go into those sadly fascinating literature. I encourage you to read on your own if you're interested in that topic. We won't take time to discuss any more of that, but certainly between the actual formation of the planet, which took place at the time of the formation of the solar system as a whole, and appearance of life, about 1 billion years went by. So during that time, of course, the Earth cooled to the point where liquid water could exist physically on the planet. And of course, I hope that you have a good understanding of how different the Earth was as an environment at the very beginning, that we discussed the fact that the chemical components of the atmosphere were completely different because at the very beginning it was much more volcanically active, a lot of volcanic outgassing. The atmosphere had a lot of methane and hydrogen and ammonia, not chemical compounds that are the primary chemicals in our atmosphere today, the two most numerous gases being nitrogen and oxygen, for example. Also during this time, there was a lot more bombardment of the Earth's surface by asteroids and meteorites and so on. So very, very different landscape back then. It is thought that the very first life forms on the planet shared some characteristics with today's prokaryotes. Why is it that we're able to put an approximate date for origin of life? It's because there are actually fossils that have been found from this time period that are thought to be traces of prokaryotic life forms from this time. Let's turn our attention now to our eon, right, the Phanerozoic. And I've now brought in another panel from the same figure or related figure in your textbook, which is to take that, just the most recent 500 million years, the Phanerozoic eon, and then stretch that out as shown here on the right-hand side. I've drawn a line to indicate where the Phanerozoic starts on this particular landscape, and you should notice a couple of different things. One is that if you eyeball what the surface of the Earth looks like, notice how different these different ovals look. The continents are arranged in different ways, and of course this is a result of plate tectonic activity or continental drift, and this has had a tremendous impact on organisms during this time period. Why? Well, that's because if you have these great big plates moving around, this will change habitats in many different ways. As a plate moves from one location on the planet to another, then it can become colder or warmer depending on where it is going. And also, as plates move towards each other, then oceans form, mountain ranges emerge, and so on. That's a little bit difficult to capture conceptually from eyeballing a static picture such as the one in front of you, which I don't believe is from your book, but it's a map of the current plates, the current boundaries, and the current directions. So you can see these two are moving away from each other. So the Atlantic is gradually getting wider and wider. And then, of course, in our own area of North America, we've got plates moving across, scraping against each other, which is causing earthquakes in this earthquake-prone area that we live in. I think it's easier to wrap your mind around plate tectonics if one sees this in a more animated form. So I've got an animation in front of you right now. This, the timeline, by the way, is up here, so you'll see this changing. This starts about halfway through the Phanerozoic, or slightly more than halfway through, because it's what? 200 million years ago. Okay, so let's go ahead and start watching this. This is an animation showing for specific points. Let me make it a little darker so you can see this better. There we are. So here, there's the clock ticking. We're at about 140 million years ago and so on. You can see these little points here are indicating sites for which there is specific data. You can see some movements. Look, we've got Africa moving away from South America here. And then watch, this is what is today's India, right? Moving in a quite remarkable manner towards Asia here, the rest of Asia, what is now Asia. And bam, right? Okay, here we are, present time. The animation ended. And of course, this interaction of the plate that India is on with the other plate with which it is colliding is what's generating the Himalayas, right? The mountain range that includes the tallest mountains on the planet. This is one of the most recent mountain ranges, and it's being formed by these two plates colliding each other, which throw the crust of the Earth into these great folds, which then are mountains. So these are obviously huge changes in habitat then, and apparently, if one was able to look at the rocks on the very peak of Mount Everest, you know, going through the snow and the ice, obviously first, and pull out the rocks, that's actually marine limestone. So what is now a mountaintop used to be part of an ocean floor. The Himalayas, by the way, are still rising. Found an estimate of about 5 millimeters a year. So that's obviously not huge changes, but over long periods of time, it can add up. So fossils are some very important sources of information about pre-existing life on the planet or more ancient life on the planet. And you may be surprised to learn that fossils are found even in our own area. This is actually a news clipping from the Orange County Register that came out a couple of years ago when a fossil whale skull was discovered in nearby Aliso Creek. And there's a little map here showing a number of different fossils that have been found. And I don't know if you can read it from where you are, but this little rectangle here says fossils found during toll road construction. And so, of course, that's how many fossils are sometimes uncovered, because great big machinery comes out and digs huge holes in the earth going down through the different layers of rock. And what happens when you're going down through lower and lower areas of rock is you're going back in time through layers of rock. And this is construction sites are very common places where fossils are actually uncovered. So at a great inconvenience to the construction crews if they have to halt for any reason. But anyway, whales obviously are an indication of a different environment from the current conditions in Aliso Creek. So just as marine limestone is now on the top of Mount Everest, we have marine creatures here in our own backyards because of the changes that have taken place in habitats as rocks are moved around during plate tectonic activity. So the interpretation of fossils is always requires knowledge then of what the conditions were like when that rock was formed as opposed to where it is right now when it's being uncovered. That's part of the interpretation. But as we look back through the fossil record, scientists analyzing fossils noticed that there was a particular period in the Phanerozoic eon which was characterized by an amazing proliferation of new life forms. This is a figure from your textbook, and if we start over here on the far left, hopefully you recognize this immediately as the Phanerozoic, right? So we're talking about the most recent 500 million years, and this is the very first part of the Phanerozoic. This point lines up with this point here. The very first period in the Phanerozoic is called the Cambrian period. This is another example of a time interval for which I am holding you absolutely responsible. Okay, but that's pretty darn easy because after all, you only need to know one eon, the Phanerozoic. It started about, what, 500 million years before present. The Cambrian period is one period. Move this out of the way here. One period within the Phanerozoic is the first period, and so you should know immediately when it started, right? This is pretty darn easy. Approximately, what, 500 million years before present, because it is the first period in the Phanerozoic. And another indication of time interval that I'll hold you responsible for is the term pre-Cambrian, which means before the Cambrian. Where is that on a timeline like this? Well, it's basically before the Cambrian, before the Phanerozoic. So here we are in the Precambrian. There we are, right? Precambrian. So Precambrian means before the Cambrian, before the Phanerozoic, and the Cambrian is the first period in the Phanerozoic. That's all the time intervals that I'll hold you responsible for. Again, I think it's more useful to just learn a couple of key locations or points that one can then use as anchors for future comparison or using it to compare additional information that you'll learn. So if we now consider why the Cambrian is described as an extraordinary time for animal diversification, this is perhaps easiest to think about in terms of what the fossil record is like before the Cambrian. Precambrian. And so the older the fossils are, or the older the rocks are, the less likely it is you'll be able to find fossils of that particular age. Information is lost over time. Plates move together, rocks are remelted in the Earth's geological activity and so on. So there certainly are fewer and fewer and fewer fossils as we go earlier, but that's not the primary reason. This thought to be why the Cambrian period is a period of such tremendous diversification. One of the oldest and most informative Precambrian fossil beds that's very close in time to the Cambrian is a area in China that has a lot of fossils that look sort of like the picture that's in front of you right now. Little tiny microscopic fossils that are interpreted to be some kind of life form, and but they're all very small, and there aren't very many of them. A bit later, there are fossils immediately before the Cambrian period. There's a particularly extensive fossil bed in Australia where these fossils are found. These are much larger. They're more complex than the earlier fossils that were found in China. But these organisms tend to be fairly small and very soft-bodied. Once you hit the Cambrian, though, and the Burgess Shale is one of the most famous, this is located in Canada, shown here, all of a sudden, for fossil beds from the Cambrian, there are huge numbers of diverse animal species that have been described, and in particular, virtually every major animal lineage that is still on the planet was found in this time period. And so even some early vertebrates date back to this time. So these organisms are much larger, they're more complex in form, and significantly there are more hard parts, meaning skeletons and chewing mouth parts and so on. So this makes a very large difference in terms of how these organisms can actually make a living. And we now have the first development of complex predator-prey relationships where animals are better able to actually consume each other rather than just simply absorb nutrients from their surroundings. So this was a very different time on the planet, and there has been a lot of interest in what the causes are for why this huge diversification event, which is considered the most spectacular example of adaptive radiation. By the way, this is a term that is in your book, adaptive radiation, meaning a period of diversification that is more rapid or more extensive than usual. The term adaptive radiation, by the way, can be applied to a local area. What's particularly noteworthy about the Cambrian is that this was a global event where there was adaptive radiation going on all over the entire planet. And so what triggered this? There are several different reasons given in your textbook, all of which have supportive evidence, and they are not mutually exclusive. So there's not one right and other wrong answers, but rather these are all presumably contributing factors for the diversification. One was the higher oxygen levels that were known to have occurred by the time of the Cambrian. What is the relevance of oxygen? Well, for aerobic organisms, oxygen then allows organisms with the capacity for aerobic metabolism then to use a lot more energy, which is very important if you're an active predator or an active potential prey item. So any kind of very active lifestyle then assumes a fair degree of burning of energy, and this is facilitated by higher oxygen levels. Complex ecological interactions is also assumed to have helped drive the diversification. Again, huge changes in relationships between organisms. Once you have these predator-prey interactions, ecologists have argued that prior to the diversification, there were a lot of empty ecological niches to be filled. There were developmental biologists point out that there was a diversification in the developmental genes around this time, which allowed for the genetic coding of more complex body forms. So there are a lot of different things that were all going on, which then presumably all contributed to this tremendous diversification which occurred at this time. So I'm not going to take time to go through the other examples of adaptive radiation in your book. I encourage you to read those and think about the differences between localized adaptive radiation, well, differences and also comparable conceptual points compared to the Cambrian. But these periods of diversification have been very important, obviously, in shaping life on the planet Earth. The flip side of diversification is extinction. And so let's now consider extinctions briefly. And so this is a figure from your book. This is figure 2814 from your textbook. And again, the familiar timeline here. Hopefully you can look at this and immediately know that you are looking at the Phanerozoic Eon, right? Because after all, here's the Cambrian period down here, and this is the present right up here. So this is the Phanerozoic. We're looking at the last, or the most recent 500 million years or so, give or take a little bit. And this graph over here on the right shows the percentage of families that went extinct. Families meaning the taxonomic unit family, which is somewhat more inclusive than species or genus, and less inclusive than something like a phylum, for example. You see this wiggly line here, which is the graph of the estimates of extinction rates. You can see that there are higher and lower extinction rates at different points in geological history. There are a couple of points in time where the extinction rate was particularly high, and these are shown in red in this particular picture, that a particularly large number of groups went extinct, meaning that they were no longer seen in the fossil record. So obviously anything that has gone extinct is only known from the fossil record. You should also note in looking at this line that there's always some background level of extinctions. This is a normal process. It's not as if it's zero and then just spikes intermittently, but the actual magnitude of the extinction is what's significant. So if we look at these 5 events then, here's the real big one, the end of the Permian. This was an extraordinary event. The estimates are that about 90% of all of the species on the planet at that time went extinct. Now, if you're looking down at where the spike is on the graph down here, you might be thinking, what is she saying, 90%? Doesn't it hit the axis there at about 50-something? Yes, it's a different taxonomic unit. This is families, so the percent of whatever that you estimate depends if you're considering species or families, because not all family has exactly the same number of species and so on. So there were some families left that had been very speciose and maybe only had 1 or 2 species left after this huge extinction event. It's really quite hard to imagine. It's clear from the rocks that date back to that time that this was a time of tremendous widespread volcanic activity, that it was a time of great heat. The carbon dioxide levels were very high. Scientists consider that volcanoes belching all over the entire planet's surface would throw a lot of carbon dioxide and sulfur dioxide into the air, along with a lot of dust, which would plunge the planet into relative darkness, which would then kill off a lot of photosynthetic organisms. Oceans were very low in oxygen. That's clear from the rocks from that time, and the sea level was much lower. So it was a time that was really incompatible with many of the life forms, and a huge number of animals, plants, and other organisms did not make it. And so some scientists say it's sort of amazing that anybody made it out of this period alive. Clearly, some life forms did, because anything on the planet right now are descendants of any of the lineages that actually made it through this highly dangerous period. Now, mass extinctions, by the way, are completely different from thinking about natural selection, because these are non-discriminant and wiping out whole major taxa. The the big one was so long ago that it's much harder to really get as much data about that extinction as about the most recent one. And the most recent extinction event, which took place on the order of about 60 or so million years ago, is the one that killed off the dinosaurs. And there's much more known about this because it was more recent, and there is a lot of evidence for asteroid impact occurring at that time. It is assumed to have some role in the extinction events. The exact role is still being debated in the scientific literature, but it's pretty clear that that this was also a time of worldwide fires and ash and darkness and huge tsunamis and other large-scale disruptive events. But that would be caused at least in part by the asteroid, possibly not only caused by the asteroid impact. But what evidence is there for the asteroid impact? Here is a figure right now. They believe that they've actually found the location of an asteroid that hit at that time, and it's located just off the coast of Mexico. And scientists that have analyzed this can even make estimates for exactly how the asteroid landed and approximately how much dust it threw up into the air and all that information. Why is it that it is considered what are other lines of evidence for the asteroid impact? One of the strongest lines of evidence is the presence of iridium. Iridium is a chemical element. It's not very common on the planet of the Earth. It is very common as part of the elemental composition of asteroids and meteorites. And so if one looks at the concentration of iridium in rocks through time, so that's this upper left panel right here, where the x-axis is iridium concentration in parts per trillion. It's trillion with a T. And this y-axis here is position in meters. This is how much geological data is reported, because as you go farther down, it's earlier. This indicates the point in time that is thought to be that period of about 60 million years ago or so. And at that point in time, there's a huge spike in iridium, and this is global. If you go anywhere on the Earth's surface and look at rocks from this time period, you see a huge spike in iridium. And notice, by the way, that the x-axis has a big chunk missing, right? We've got 0 to 100 here, and then we jump to 3,000. So the magnitude of this spike is very, very much more concentrated than normal, and this, because this spike is shown worldwide, the interpretation is the asteroid hit, was immediately vaporized, ended up in ash that then circulated in the atmosphere all over the entire planet, and then over a period of some years, then rained down and collected in the sediments worldwide, which is why we see this spike everywhere at this particular time. So it's pretty clear an asteroid hit. There's also evidence from particular kinds of minerals that formed at that time. So, okay, let's switch gears a little bit now and talk about prokaryotes, again with the backdrop of a geological timescale, and the origin of life is about 1 billion years after the formation of the planet, so about 3.5 billion years before the present time. And it is thought that the very first life forms on the planet had shared at least some characteristics of today's prokaryotes, today's bacteria and archaeans. So before we talk about those characteristics, let's first consider that there is a growing appreciation for the importance of and the intimate associations that we have with microbes, particularly bacteria. This has been in the news a lot. Here's a fun posting. I put this on the website as optional reading. This figure here shows an example of what happens if you take a bunch of humans, shown here in the middle, whether it's the front surface or the back surface, and swab the human in different locations, and then look at the makeup of the bacterial species that inhabit different parts, and then analyze different humans. The purpose of such studies are to try to get an idea of, do different people, are they colonized by different groups of bacteria? Or how does the diversity in bacteria that live on our skin relate to location in different humans. And one of the interesting things that has been found is that different locations on a human's body tend to be characterized by particular communities of bacterial species. So if you compare swabs from the armpit versus the forearm, between the toes, forehead, up the nose, all these are different microhabitats for these different groups of bacteria. So what these little pie charts show is how many you have of the different kinds, where the kinds are all listed over here. That is, if you take a couple of different humans, the bacteria in their armpits of the different humans will be more similar to each other than they will be the same human comparing, say, the armpit and the forehead. So so scientists now think about these as different microhabitats, much in the same way as we think of different habitats on the planet of the Earth, desert versus mountain ranges and so on. So the armpit and the forehead are as different to microbes as deserts and mountaintops are to us, and so on. Anyway, it's quite clear that we are outnumbered by the microbes that are associated with us. So for example, if you take a human being and figure out how many cells are in that human being's body, and then compare that to the number of bacterial cells associated with that human's body, meaning in the digestive tract and on the skin, for example, which are the two primary locations, human cells are outnumbered about 10 to 1. So we're carrying around very large numbers of bacteria on and in our body. But this isn't just a bunch of passive hitchhikers. It is now very apparent that they have tremendous effects on our health and our physiology. And so here's just one example of many. Again, I posted this as optional reading. I picked readings, by the way, that are very easy to read and have nice graphics. I'll just read the first sentence here. Being caked in germs sounds unpleasant, but friendly bacteria living on our skin may have the vital role of keeping in check inflammation triggered by injury and unwanted bacteria. Okay, so a growing appreciation for a positive role and an important role that some kinds of bacteria then have if they're on our skin. So in fact, this is one of the reasons why there is a growing discomfort with the idea that our soaps are antibacterial. And if you go into any store and check out the soaps, it's quite common to see antibacterial, and so on many of the products on the shelves. And so these specifically have antibacterial chemical compounds in them. And there is some concern that this may not be a good idea, either because this may take out the so-called friendly bacteria that have a positive physiological or medical role in human health, or also potentially causing problems with the spread of resistance of bacteria to antibacterial chemical compounds. We're not quite at this situation here, but prediction is, in the future when we go check out our soap, we'll see labels like this, now full of bacteria, right? Where instead of being antibacterial, they'll be like pro-bacteria, and our soap has better bacteria than the bacteria in your soap, and this sort of thing. Anyway, well, I'm sure we'll see that. By the way, I don't want you to avoid soap, okay? That is not the message here. I'm not anti-soap at all. In fact, any reputable medical establishment, so the Center for Disease Control of the National Institutes of Health, commonly list hand washing with soap thoroughly is one of the major things that helps prevent or decrease the spread of serious diseases. So keep washing your hands. You just might want to avoid antibacterial soaps. So let's now consider some of the characteristics of bacteria and the other major prokaryotic group, the archaea. And so the first thing we should consider is their relationship and their evolutionary history, which is summarized in this figure from your textbook, which shows the relationship between the three domains, which hopefully feels like review to you, because after all, we discussed the work of Woese and his collaborators who used sequences from ribosomal RNA to look at the phylogenetic relationships of all living organisms and found that they came out very clearly in three major groupings, two of which are prokaryotes, as shown here. The bacteria and the archaeans are both prokaryotes, and the eukarya, the group we're in, the domain we're in, is the only group that is the eukaryotes. So recalling that time is proceeding from left to right, then there was an early split between bacteria and the other lineage that led to today's archaeans and eukaryotes, as shown here. So we are more closely related to the archaeans than the archaeans are to the bacteria. So what do we need to know about bacteria and archaea and eukarya? Well, here's a handy table that summarizes just a very tiny handful of distinguishing characteristics of these three major domains. So we've got bacteria and archaea and eukarya. I think students find tables very hard to wrap one's mind around, and that's not just students, it's sort of people in general. So I would encourage you to think of putting these two pieces of information together, namely this phylogenetic tree with this table. What do I mean? Let's go ahead and walk through this together. We've got 1, 2, 3, 4, 5, 6, 7, 8, 9 different characteristics that are highlighted in here. Let's go ahead then and see how these map out on the phylogenetic tree, starting with number 1. Number 1 is the DNA enclosed by a nuclear envelope? And it should not surprise you at all that the answer is no for the two groups of prokaryotes, and it's yes for the eukaryotes. After all, that is the distinguishing feature of a eukaryote, right? So if we go back here, what does that mean in terms of where a nuclear envelope evolved? Well, clearly it happened in the lineage. It is most parsimonious to assume that this occurred in the lineage of the eukaryotes. I'm putting a number 1 here to show that it corresponds with number 1 on this list, right? So here we have some point in the lineage that led to all eukaryotes evolution of a nuclear envelope, which did not occur in the other lineages. Moving on to exciting fun fact number 2. Circular chromosome present, yes in bacteria, yes in archaea, although in some bacteria it's linear, but in eukaryotes it's all linear. So clearly if we go back to this one then, somewhere in this lineage there was a transition from being circular to linear. And again, it is more parsimonious to assume that it occurred in the lineage that led to the eukaryotes than it occurred earlier and then was lost in other branches. Moving on to number 3, organelles enclosed by membranes present. No, no, and yes for the eukaryotes. Looks like number 3 is here as well. Now notice, by the way, or I should point out, by the way, that although we've got 1, 2, 3, that in no way is a statement of temporal order. That does not mean that number 1 had to have happened first before number 2 and so on. We actually don't know in what specific order these changes took place in the evolutionary history of this group. They just all clearly happened early on in this branch, but we don't know the temporal sequence. Okay? Number 4, rotating flagella present. Yes, yes, and no. In eukaryotes, they undulate. What does undulate? That just means they wave back and forth as opposed to rotating. This may sound like an amazingly picky thing, but in fact, there's more to it than it just simply appears on the table. Not only are the motions of the flagella different, but the entire biochemical machinery and the proteins that make up the flagella are arranged in very, very different ways. So huge changes in the flagella relative to bacteria and the archaea. It's not just how they move, it's everything else about the flagella as well. So completely distinct structures, very, very different. Number 5, multicellular species, no, no, yes. So clearly that's something else that evolved somewhere around here, so we'll stick 5 on here too. So we've got this whole arrangement here, kind of building up on the eukaryotes. That's just a artifact of the way in which they are ordered on this table. Number 6, plasma membrane lipids composed of glycerol bonded to unbranched fatty acids. The answer is yes for everybody except the archaea, which are different. They're the only ones with these unusual, highly branched lipids in their cell membranes. So this is something really different about the archaea. So let's go ahead and put that in right here. These are the branched lipids in the cell membrane. This is completely different from the other organisms, very distinctive of the archaea. So this is what? This is a synapomorphy for this group, right? It is a shared derived characteristic that unites the archaea. It distinguishes them from the other groups. How about number 7? Cell walls, when present, contain peptidoglycan. The only one of the 3 domains that has this is the bacteria. So going back here, this is a distinguishing feature. of the bacteria. So we have peptidoglycans in peptidoglycan. There's a particular kind of chemical compound in the cell walls of bacteria. Very unusual, very distinctive for bacteria. It also means that if bacteria are being targeted chemically, then one of the ways that scientists choose to target bacteria is, of course, by some characteristic that is not shared with other groups. So anything that targets peptidoglycans or their biochemical synthetic metabolic pathways then is something that for which bacteria would be vulnerable and the other groups would not. Number 8. RNA polymerase composed of more than 10 subunits. No. Yes, yes. Now we see something that actually joins the archaea and the eukaryotes. So that's something then that presumably occurred here sometime. So RNA polymerases became larger, they became more complex. And this is a greatly simplified table, by the way. There's not just one, there's a large number, but there are complexity and size of RNA polymerases are very distinct in these two sister groups, the archaea and the eukarya, that are not shared by the bacteria. And number 9, translation initiated with which amino acid? Is it methionine or is it a modified form of methionine? Again, this is something that the archaeans and the eukaryotes share. So this is another issue here. So I hope taking time to go back and forth between those two will also encourage you, both with this group but also with other groups. If you're trying to understand the evolutionary history of a group, it's often very helpful to take a phylogenetic tree and go ahead and superimpose evolutionary changes on it, because after all, once something occurs in the lineage, then it is shared by all the descendants in perpetuity, unless, of course, it is lost by some descendant lineages, which, of course, is possible. But it makes it easier to wrap your mind around what things are shared and what things are different if one does it in an explicit phylogenetic context, such as just done. Now, hopefully you understand the logic of where I place these. Of course, a good way for me of telling how good you are doing this is doing a quick clicker question. So let's go ahead and do this. We'll do answer counts, meaning you get more credit if you answer this correctly. I would consider this extremely easy. I want to see like 99% getting this right. So here we have the phylogenetic tree for the 3 domains, and I'm providing it to you, although I would certainly expect you to actually be able to draw it if it was a blank screen. But anyway, here they are, and here's a fact. Sexual reproduction is seen in eukaryotes, meaning fusion of gametes, sperm and egg fusing together, but not in the prokaryotes. On the phylogenetic tree below, which number indicates the likeliest point for the evolution of sexual reproduction then, if this is just seen in eukaryotes? 1, 2, 3, 4, or 5? Not sure? Good time to chat with your neighbor, pick their brains. I'll give you 15, 20 seconds or so to compare notes, and then we'll see what you guys think. Okay, everybody should have figured this out and clicked in, right? Okay, it's stabilized. Sort of. Sheesh. Okay. Let's see. Awesome. But only 92. What's with 8% of you? Sheesh. Okay. Let's see. So, if this is something that's seen in eukaryotes, then hopefully you thought, okay, where are my eukaryotes? They're in the eukarya, right? That's where they are. If something occurs just in this branch and it's not shared by the other ones, then this is the most logical location. If it was here, for example, then this should be shared by the archaeans unless, of course, it was lost, which would be a less parsimonious explanation. So, the 8% of you who got that wrong should spend a little time working with phylogenetic trees and thinking about placement of evolutionary changes on phylogenetic trees. Okay, so continuing thinking about reproduction here. Go ahead and start up the movie so you can visualize it while we're chatting about points here. So, In these prokaryotes, reproduction, one thing to keep in mind, these organisms are in fact haploid. They do not have sexual reproduction in the sense of a sperm and an egg fusing together as gametes, which is something you see in most eukaryotes. They do reproduce by, of course, DNA replication, and then a form of cell division called fission specifically. And so you can see in this group here, one will divide into two, and then each one of those will divide into two, and so on. In addition to the DNA that's in their circular chromosomes, they do have extra loops of DNA called plasmids, and they do have behaviors for exchange of genetic material, specifically plasmids, as are seen, and are given a specific name of conjugation. This is typically one way where a plasmid is exchanged from one individual, is passed from one individual to another. It's not usually a reciprocal exchange of genetic information. So very different than what you see in most eukaryotic cells. I often get questions about how fission is different from mitosis. After all, if you can compare fission and mitosis, in both cases you have a cell dividing into two identical daughter cells. A huge difference, of course, with a cell undergoing mitosis. A eukaryotic cell is that because there's a nucleus, the nuclear envelope has to disintegrate and then reform, and you have the formation of a spindle and so on. So there are a number of different additional steps. It's more complicated procedure in a cell with a nuclear envelope. But in both cases, you certainly have replication of DNA followed by cell division. That is a common feature. When you think of bacteria, you probably think of diseases. After all, they have important positive roles in health, but also they do cause a lot of diseases which have a negative impact on humans. There's a partial list in Table 29.2 in front of you. You should certainly look at these. Do you have to memorize them? Of course not, but it should be of interest to you how many important major killers are in fact bacterial diseases, pneumonia and syphilis and tuberculosis and leprosy. One of the main ways in which bacterial diseases are fought in a medical sense is by treatment with antibiotics. By definition, antibiotics are chemical compounds that kill or slow the growth of bacteria. Antibiotics typically target issues of bacterial biochemistry or physiology that are not shared with other kinds of organisms. I'm going to go ahead and skip across gram-negative and gram-positive bacteria. I think that's pretty easy for you to go ahead and read. But I want to pass to the next topic, which is diseases caused by archaeans. I do expect you to memorize every single disease caused by archaeans. Yeah, haha. Okay, somebody got the joke, yes. There is, in fact, no table in your textbook. Why? Because it would look like this. Okay, they're not going to waste a textbook page on this. Astonishingly, this large, diverse group is not associated with any disease. There is a mention in your textbook of a bit of a smoking gun that was identified in 2004 that has since been repudiated. I put some optional reading on the class website for you. In fact, there is no well-established disease. The smoking gun that people were all excited about in 2004 was some scientists who were studying the microbial composition in the mouths of individuals that had gum disease found that they found some archaeans in there. It was like, yes, we finally found a disease that these microbes cause. Yes. But when the data were examined more systematically and thoroughly and more cases were examined, it was pretty clear that the archaeans were only present in between about a quarter and a third of the patients. There are many, many other microbes present, and so one more minute, guys. So the standards for a positive identification of a relationship between a pathogen and a disease were in fact not established. One of the ways in which microbes, though, are very known for is the tremendous metabolic diversity that they have compared to eukaryotes, which are much more limited in our metabolic diversity. And so what is meant by metabolic diversity? That section of your textbook, by the way, goes into a lot of detail. I would say go ahead and read it because it helps you understand the broad patterns, but focus on the broad patterns, namely the ways in which organisms can produce ATP from different sources of energy and the ways in which organisms can utilize different sources of carbon.